This is Dr. LAILA JARAGH-ALHADAD from Kuwait University. I'm glad to have this Best Research Award and International Research Award on Oncology and Cancer Research 2021. My title is Development of Nimesulide Analogs as Dual Inhibitor Targeting Tubulin and HSP27 for Treatment of Female Cancers. So we want to highlight that we synthesized the nimesulide analogs, characterized them, confirmed the structure for L4, and we run a Western blot MTSA to show the compound's potency. To move from nimesulide as a fever drug to anti-cancer agent. So this is the graphical abstract to show the synthesized compound L4 and how we used Western blot assay and MTSA. So let's start with our abstract. In the drug repurposing paradigm, we used to treat specific disease by a medicine that we can use it to treat another disease also. So we start from nimesulide, which is anti-fever drug targeting COX-2. So it has anti-inflammatory effect. And we want to prove that it has anti-cancer effect also. So our targets is tubulin and heat shock protein 27. These are important in cancer therapy, important targets because they both are expressed and elevated in cancer cells. In this regard, we start from the nimesulide skeleton, as you can see in this chemical compound. We start from the nimesulide skeleton and we change different ligands, and we would like to characterize that and see the biological activity. When we said characterize that, it means we used proton NMR, carbon 13 NMR, IR, melting point, DSC Thermal Analysis, and we confirmed the molecular weight by GCMS. So our first target is tubulin. It's important for any cell to function, for cell division, movement, intracellular trafficking, for shape maintenance, and for cell proliferation. You can see in this picture that alpha and beta tubulin dimer, when it binds together, it forms microtubules. These microtubules are important for the mitotic spindle, and the one cell will give you two identical daughter cells. So it's important for cell division. So all cancers elicit high dependence on tubulin polymerization and depolymerization for cell division. And they found that there is microtubule inhibitors targeting these microtubules, but after initial treatment, these inhibitors becomes less effective and developing a drug resistance. So further studies required to discover new tubulin inhibitors as anti-cancer agent within a minimal side effect and ideal therapeutic index. Our second target is HSP27, which is important for refolding the damaged protein. So studies found that HSP27 is highly expressed in cancer cells, including liver, bladder, pancreatic cancers, ovarian, and breast cancers. Some research studies targeting HSP27 and in the clinical trials using antisense, second studies using antisense combined with chloroquinone, and one new study they use OGX, okay, to target HSP27. Our newly published research is this compound. It's a nimesulide analog where we conjugated to cholesterol encapsulated into low-density lipoprotein to target both HSP27 and HER2 receptor through HER2 pathway. And the IC50 values were really promising. In the drug repurposing strategy, it has a considerable attention in these last few years. It offers the disease treatment at reasonable cost using medicine already administered to patients. And the conventional cancer treatments is chemotherapy, kinase inhibitors, and monoclonal antibody. However, this medication restricted by high toxicity, non-selectivity, and drug resistance, and low therapeutic index. Therefore, the development of tumor-specific agent should be considered. And we will talk now about our objectives, which is we start from nimesulide skeleton to design new analogs, then characterize them, and biologically evaluate them. So let's start from 2-amino-5-nitrophenol. This is the main skeleton, which is in red color. And we add first ligand in position A, which is fixed in all these 4 compounds with 2,5-dichlorobenzoyl moiety. Position number B and C, we put a methyl group, also it's fixed in these 4 compounds. Only position D with different moieties, 4-methoxybenzoyl benzamide, 3,4-dimethoxybenzamide, 4-iodobenzamide, and 1-naphthamide. So I have to notice that all chemicals for the chemical analysis for biological evaluation, it's from Sigma Aldrich, and all the tests were done at Kuwait University Research Sector Project Unit, RSPU, and I would like to acknowledge them. So we start the scheme, the first synthesis from 2-amino-5-nitrophenol, and to receive compound A. Compound A, it has fixed position with 2 and 5-dichlorobenzoyl chloride. And then we converted this NH2, substituted with the sulfonyl group. After that, this compound B, we methylate this hydrogen. And in compound C, what we did is we reduce this NO2 group to NH2 group. So this is the first scheme. The second scheme, we start from compound D, which is in the middle. And when we added 4-methoxybenzoyl chloride, we receive L2. So we named the compound L2. And when we added 2,3-dimethoxybenzoyl chloride, we received compound L3. And with the addition of 4-iodobenzoyl chloride, we receive compound L4. And with the addition of 1-naphthalamide, we receive compound L5. So we have 4 compounds, L2, 3, 4, and 5. We confirmed, we chose one compound, L4. We confirmed its crystal structure by X-ray crystallography technique, which has a clear colorless block, and it's adapted triclinic system with B1. And we summarize all the X-ray data collection and structure solution parameters in these tables. And you can find here also the summarized bond angle and bond length. After that, we run the proton and carbon 13 NMR. So for proton NMR, you can see we run it in CDCl3, and I will show you for carbon and proton NMR the graphs. We summarize the figures in this graph, in this table, and you can see in this table both all L2, L3, L4, L5 compounds has this NH peak after it was NH2. So this is proof that the reaction happened. Okay, and instead of the NH2 in the chemical structure, we have now the peak for NH. So the substitution happened. You can see here, this is proto NMR. You can see the lower shifts for the methyl groups and the higher shifts for the aromatic groups and for the NH peak. And this is the carbon NMR. The IR bands, we summarize them on this table for the compounds L2, L3, L4, L5, with which one is broad and sharp. You can see the figures here and all the details in the paper on the structure and the structures in the paper exactly. So we confirmed the mass also. You can see for L2, the mass which we found is 508 and the calculated one is 509. So I compared between every single compound, the found and calculated masses, and this is confirmed. L2, L3, L4, L5 mass, the molecular weight by GCMS. So also we run the DSC Thermal Analysis, where it showed you the exothermic peak, endothermic peak, and how and monitor by the binding interactions involved the biological macromolecules. So we have the ligand, so the compound L2, L3, L4, and 5 shows different behaviors. They have sharp endothermic peaks, varies between 150 and 320 degrees Celsius, and broad exothermic peaks varies between 400 and 550 degrees Celsius. So the first endothermic peaks correspond to the melting of the ligands, while the second one represents the decomposition to different species. You can see here in these graphs, okay, the first peak on the left, which is for the exothermic, and on the right, which is for the endothermic peaks. So what we did after that, we ran Western blot. Western blot, we used SKOV3, ovarian cancer cell line, and SKBR3, breast cancer cell lines. So from the confluent plates, we harvested the cell, we lysed them, we run gel electrophoresis, and we transfer the gel into the nitrocellulose membrane, and we block it with non-fatted dry milk to target these two proteins, HSP27 and tubulin, and with primary and secondary antibodies. And we used beta-actin as our housekeeping control. So you can see from this Western blot that SKOV3 and SKBR3 cell lines expressing HSP27 and tubulin proteins. And you can see the control is beta-actin. After that, we run MTSA. We wanted to follow the yellow MTT dye and its reduction. So first day we seeded the 96 well plates, and second day we treated with our compounds at different concentrations. We kept it for 72 hours, and then we measure the absorbance at 570 nanometer, and the results and the data were normalized to controls. We summarize the data in this table. So this is the major structure, and this is the position D, where we have L2 with 4-methoxybenzamide, L3 dimethoxybenzamide, L4 4-iodobenzamide, and L5 1-naphthalamide. We found that the IC50 values to inhibit the SKOV3 cell line in micromolar range, it's between 0.23 and 2.02, while the IC50 values to inhibit the SKBR3 cell line in micromolar range also, it's 0.5 till 3.73. It's potent and it's good IC50 values, but it shows the potency in SKOV3 more than SKBR3. So someone asked me why you chose these two cell lines for ovarian and breast. So these cell lines have high expression of HSP27, So because of that, we use them. And we found now the first data showed us that these compounds are potent in SKOB3 cell line, ovarian cancer cell line, more than SKBR3. One thing to notice that, to mention, it's the statistical analysis done by graph pad PRISM software, and the IC50 values were normalized using non-linear regression analysis. So in depth, I put here the compound structure, and you can see L2 and L4, it has lower IC50 values more than L3 and L5. You can see that L3 and L5 has positions on the ortho and meta position, the ligand substituted in ortho and meta position, but L2 and L4 has the potent activities because of the para position. We summarize from that the compounds are potent when it has substituent on the para position and has biological activity. So by that, we conclude that our nimesulide analogs were synthesized and catalyzed, characterized, and biologically evaluated for their potency. L4 was confirmed by X-ray crystallographic technique, and the IC50 data revealed that our agents are potent and were able to inhibit tubulin and HSP27 function in both surrogate model SKOV3, SKBR3 cell lines and the micromolar concentration. And we hope in this near future to synthesize new ligands to improve the biological activity in the nanomolar level. I would like to acknowledge, to thank our collaborators in King Saud University in Saudi Arabia, Dr. Jamal Ad-Din Harisa and Dr. Faris Al-Unizi. And I would like to thank also Kuwait University Research Sector to approve our grant, which is SC 1418. Also RSP unit in Kuwait University to provide us with all facilities. Thanks, and we will stop recording here.